Namaste. A short while back, tigers were captured from Kanha Tiger Reserve in Madhya Pradesh and Bandhavgarh Tiger Reserve in Madhya Pradesh, taken to Satkoshia Tiger Reserve of Odisha and released there. Now, why would we do such a thing? In this lecture, we are going to look at reintroductions and outbreeding, and we'll see what are the scenarios in which we need to move these animals from one place to another place, and what are the considerations that are required when we are doing such an activity. So, let us begin with some definitions. The first one is translocation, deliberate and mediated movement of wild individuals or populations from one part of their range to another. Example, movement of tiger from Kanha Tiger Reserve to Satkoshia Tiger Reserve. So, essentially whenever we are doing a deliberate movement of wild animals, wild individuals or populations from one part of their range to another, we call it translocation. So, translocation is basically movement of animals from one place to another. Next is reintroduction, an attempt to establish a species in an area which was once part of its historical range, but from which it has become extinct. Example, movement of tiger from Ranthambore Tiger Reserve into Sariska Tiger Reserve following the Sariska debacle. Now, the crisis of Sariska is something that we are going to discuss in our 8th module, but essentially what had happened was that in a tiger reserve in Rajasthan that goes by the name of Sariska Tiger Reserve, all the tigers were hunted out by poachers. Now, Sariska happened to be a place that was a range area of the tigers, so tigers were naturally found there. But then this area, after the loss of all the tigers, it became categorized as a historical range, because it was earlier a range, but there are no longer any tigers. So, we will call it a historical range. Now, reintroduction is an attempt to establish a species in an area that was earlier a historical range in which there are no more animals present. Now, why do we do such a thing? Because if we know that that certain area was a historical range, then it means that this area was suitable or is still suitable for the survival of this particular species. So, this is a good area in which we could establish a population of the animals, and so we try to reintroduce the animals. So, reintroduce because it was earlier there, now it is extinct. So, now we are introducing it once again into the system. So, this is known as reintroduction. Next, we have reinforcement or supplementation. So, in the case of supplementation, we have an addition of individuals to an existing population of conspecifics. So, conspecifics means that you already have a population of the same species, but you are now trying to add more individuals into this population. Example, movement of tigers from Pench Tiger Reserve to Panna Tiger Reserve. So, we already had a population of tigers in the Panna Tiger Reserve, but the population was less. So, we try to reinforce that population. To supplement that population, we brought in more tigers from Pench Tiger Reserve into the Panna Tiger Reserve, so that the population in Panna Tiger Reserve could grow up. And the fourth thing is introduction, an attempt to establish a species often for the purpose of conservation outside its recorded distribution, but within an appropriate habitat and ecogeographical area, and this is often used when there is no remaining area left within a species historical range. So, basically what we are doing in all four of these is that in the case of translocation, you have an animal in its range, you are taking it, you are moving it to another area in range, or essentially, or say some other area. So, basically when we say translocation, you can just remember that it is movement of an animal or a population of animals from place A to place B. So, let us call it place A to place B. So, any such movement will be translocation. Now, this translocation can be done for introduction or reintroduction or supplementation. So, translocation is the process and these are the three reasons. So, let us call it a process and these are the reasons why we are doing this translocation. Now, if we look at these reasons in more detail, so let us look at introduction. So, essentially you had this big area that was earlier a range for a species. So, let us call it a species A range. Now, this was a historical range and this is the most extensive range that this species ever occupied. But then later on it so happened that all of this portion was then taken away, say for farming or say for other developmental activities or say for pastoral land. So, now all of this area is now gone. So, in the case of introduction, what we will do is that we will take some other area that is outside of the historical range, but it has suitable conditions, and then we will try to bring the members of this species into this area. So, now we have species A in this area, but then this area was never naturally or historically a range for this species. Now, an example could be, say we have equatorial rainforest in Kerala. Now, in state of Kerala, in the equatorial rainforest, suppose we have some species of frogs that are found in that area. Now, if the forests are under a huge anthropogenic pressure, so people want to get into those forests and people want to chop off those trees and maybe convert it into some other land use. So, in that case all of its historical range, because it was an endemic species to Kerala, so all of its historical natural range would be gone. So, we could look for a place that has a similar climate. Now, a place with a similar climate would be in the northeastern part of the country. So, let us say that there is a small patch in Assam. Now, that small patch in Assam never had this species of frog, but then we have calculated that the soil types of both these areas are same, the climate of both of these areas are the same, and the other species that are found in both of these areas are nearly similar. So, we have those tall equatorial rainforest trees that are found in Assam as well. So, then we could take some of these animals from Kerala and then put them into the state of Assam, into our designated area. So, this activity in which we are taking out an animal from its range and then placing it into an area that was never its historical natural range, so that we can establish another population there, or because its original habitat is now been taken away because of some other purposes, such a process will be known as introduction. Introduction because the animal is being introduced there for the first time, it was never present there before. The second thing is reintroduction. So, in the case of reintroduction, you have an area that has species A. Now, it was naturally found in this area, but then say there was some activity, say poaching, and so now you have this area that is now devoid of species A. So, you bring species A from some other area, so that you again have species A in the same range or the same habitat. So, such a process will be known as reintroduction, because it is reintroduction because the species was already there in some historical time, now it is no longer present, and so you are reintroducing it in this area. So, this is reintroduction. And third is supplementation. Now, supplementation, as the name suggests, so you have this area in which you had say 10 individuals of species A. So, you add 10 individuals of species A from other area, so that now in this same area you have got 20 individuals of species A. Now, when we are doing such an activity, We are just trying to bolster the number of individuals that are there in this small area. We are trying to supplement individuals into this area, and so this is known as supplementation. Now, it is easy to understand why we are doing an introduction, because there is no other area left for this species. It is also easy to understand why we are doing a reintroduction, because this place is now devoid of the animals. But then why do we want to do supplementation of an area? Or even in the case of reintroduction, when you are adding species A into this area, then probably in the first instance you added say 2 tigers into this area, or 2 individuals of species A. So now you have 2 individuals, and then later on you add 6 more, so that you have now 8 individuals of species A. So in the case of reintroduction, why don't we stop at say 1 individual or say a pair of individuals? So you have a male and a female. Why do we do that? Why do we reintroduce quite a substantial number of individuals, maybe in different stages? So here we are talking about the concept of genetic rescue. So if you have a very small population and most of the individuals become related to each other, we might start seeing the impacts of inbreeding that we discussed in a previous lecture. Now, genetic rescue is the restoration of genetic diversity and reduction of extinction risk in small isolated inbred populations, often through natural or artificial movement of individuals that bring in new alleles to the gene pool. So we understood the extinction risk in the previous lecture of population viability analysis. So in this case, what we are trying to do is that if there is a small isolated and inbred population, so in this case there is a huge amount of extinction risk that is present with this population, because there is a very little amount of genetic diversity, there is a high amount of inbreeding depression, and maybe a number of recessive diseases are showing up, or any other infections that prop up into this population flare up very easy. So because of which we have a huge amount of extinction risk. Now we are trying to reduce this extinction risk by restoration of genetic diversity. So we want to increase the amount of genetic diversity that is there in this small population. And how do we do that? We could either go for a natural movement of individuals or an artificial movement of individuals through translocation, so that we have new alleles in the gene pool. Now, natural movement of animals is something that we saw before in the case study of the Isle Royale wolves. So in this case, the wolf population was strongly inbred, but then in 1997 one individual came in. So in the period before 1997, so say in the last 15 or 20 years, we were seeing a huge amount of inbreeding depression in the population and a large number of recessive disorders in the population. But then once we had this one new guy that came in, we started seeing a massive reduction in the genetic disorders that were seen in this population. So this one individual, just by bringing new alleles into the population, and it was able to provide a genetic rescue of this small population. So this population was as small as say around 10 individuals. So there were these 10 individuals which were highly related, and just one new individual that came in was able to provide a good amount of genetic rescue by increasing the amount of variety that was there in the gene pool. In case this natural movement is not possible or is not feasible in a short period of time, we could go for a translocation of animals. So for instance, in the case of say Panna Tiger Reserve, if we had a small population of tigers there, then it is possible that some other tiger from Bandhavgarh may have come into the Panna Tiger Reserve while it was dispersing out. But then that would have taken a very long period of time, and in that period our Panna population would have become further inbred. So to rescue that, we just brought in more individuals from the Pench Tiger Reserve. So such an activity would tell us why a translocation is important. But then when you are bringing in an individual from outside population, how diverse should the rescuer be? So here we come to the concept of outbreeding. So inbreeding is bad, but then outbreeding can also be bad. So outbreeding is the practice of introducing unrelated genetic material into a breeding line. Now when you are bringing in unrelated genetic material, so which means you are bringing in individuals that are different from your population, it could have two impacts. It could have the impact of outbreeding enhancement or outbreeding depression. Now outbreeding enhancement means improved biological quality in a hybrid offspring. Now, why would you observe an improved biological quality in a hybrid offspring? Say all your populations were having, say, some genetic disease, some recessive disorder that was rampantly available in the whole of the population. Now, if you have an outbreeding with a new genetic material individual, then the amount of recessive genes that are present in the individual would go down, because your unrelated individual would be bringing in some new alleles which may be dominant, and then that would bring down the impacts of the recessive alleles that are already present in the population. So, that phenomenon goes by the name of outbreeding enhancement, and it is also known as hybrid vigor. So, this is very commonly seen in the case of plants. So, for instance, if you have a clone of maize that grows to say this height, so you have two clones. So, this is clone A, this is clone B. Now, all the individuals in clone A have been grown by inbreeding amongst different individuals of this plant of maize, so that we had some desired characteristics. So, probably this clone A shows good number of seeds and probably a fast growth. And in the case of clone B, probably it has resistance to disease and also resistance to drought. And now, if we breed both of these together, then the progeny that comes up, it would have, say, both of these good qualities. So, it would have good number of seeds, it would have faster growth, it would have also resistance to diseases and resistance to droughts. So, essentially any individuals of clone B did not have these good qualities, any individuals of clone A did not have these good qualities, but then this hybrid has both the good qualities inside it. So, this is something that would go by the name of outbreeding enhancement or hybrid vigor. And in most of the situations we have also observed that these outbred individuals, they also have greater amount of resistance to diseases, they also have say physically a very magnificent body, they are also in most cases they grow taller and they have other such properties. So, these things go by the name of outbreeding enhancement because of some amount of genetic rescue that is happening. But then there is also another thing that could happen, which is outbreeding depression, which is decreased fitness in a hybrid offspring when compared to parents or to offsprings from crosses that are between more related individuals. Now, you could ask that we can understand why an outbreeding enhancement would occur, because we are getting good qualities from both the parents. But then in the case of outbreeding depression, why would that happen? Well, that could happen because you are getting bad qualities from both the parents. So, it is also possible that in the case of our hybrid individual, this individual does not get good qualities from here, it gets the bad qualities from here and it gets the bad qualities from here. So, in this case, these individuals of clone B, they had also the properties of poor number of seeds and slower growth, and these individuals of clone A were having the property of disease and drought susceptibility. Now, it is also possible that when we talk about this hybrid offspring, it gets these bad qualities from here and it gets these bad qualities from here, in which case it will be unfit or less fit as compared to both the parents. So, this is what is outbreeding depression, a decreased fitness in the hybrid offspring when compared to the parents or to the offsprings from crosses between more related individuals. Because in the case of the parents here, so these parents were having these two good properties, these parents were having these two good properties, but then this hybrid offspring that came up, it did not have any of the good properties from anywhere, it came up with all the bad quality. Now, in the case of wildlife, outbreeding depression manifests in two main ways. One is that you have intermediate genotypes that are not adapted to either parental habitat or there could be a breakdown of biochemical or physiological compatibility in the hybrids. So, for instance, coming back to the drawing board, when we say that the intermediate genotype is not adapted to either parental habitat, what we are saying here is that in the case of our clone A, it was growing in those areas that did not have diseases and which did not have drought. Now, this individual that comes up, clone A was also growing in areas which required faster growth because it had more number of competitors, say weeds in that area. Now, your clone B was growing in those areas where it did not have any competition, but it had quite a number of diseases and it was also in an area that was suffering from drought. Now, this individual that comes up, this offspring, it is susceptible to disease, it is susceptible to drought, it gets those qualities from clone A. So, you cannot place it in an area that has clone B because it will not be able to survive because it does not have those good qualities. Similarly, it has poor number of seeds and slower growth from parent B, so it will not be able to survive in the habitat of parent A. So, what we are saying here is that you have intermediate genotypes that are not adapted to either parental habitat, or it could also happen that when we are talking about the breakdown of biochemical and physiological compatibility in the hybrids, then it is possible that these hybrids that come up, so probably in this area we were having, say, fertilization by large insects. So, it required a larger size of flowers, and probably in this case we had fertilization by slender insects. So, these were insects that were more thin. So, in the case of the hybrid, it comes up with a flower. So, in the first case we required those flowers that were like this big. So, in that case you required flowers to be big so that larger size insects can get inside. On the other hand, in the second case you required flowers that were long and that were slender, so that your insects were able to get inside. Now, it is possible that in the case of the hybrid, it does not have smaller flowers, it does not have larger flowers, it has flowers that are intermediate. So, in the case of these intermediate flowers, it is possible that your larger size insects are not able to get inside, and at the same time your slender insects are not able to get inside. So, that would lead to some amount of biochemical or physiological incompatibility between the hybrids. Now, let us look at two case studies. The first case study is that of ibex. Now, ibex is a goat like wild animal, and by the 1850s alpine ibex, that goes by the specific name of Capra ibex, was extinct in the high Tatra mountain range at the Slovakia-Poland border. So, it was a goat that was found in the high mountains. Now, this species was reintroduced in 1901 from Austria, but the number of specimens that we could get from Austria was less. So, to increase the genetic diversity, because people thought that we have less number of individuals, there will be more amount of inbreeding depression. So, let us increase the genetic diversity. So, what they did was they also brought in individuals of Capra hircus and Capra nubiana species that were introduced. Now, in those days it was believed that Capra ibex is a goat that is found in the alpine areas, and these are also the same species, they are just the subspecies of the same goat that are found in more hotter regions. So, what happened was that in a short while all the animals died again. Now, what was the reason? In the case of Capra ibex, in the case of these animals that grow in very cold areas, so you do not want to have fawning or childbirth in the very cold climates, because in those times we would be having very high amounts of snowstorms that are coming in that area. So, because the offsprings are more susceptible to cold, so they would die off. So, in the cold climates we had that this species was rutting in winters, so it was mating in winters and it was giving out the offsprings in spring. The other two subspecies at that time, or species now, they come from more warmer areas, so they have mating in the autumn season and they fawn in winter. because they are coming from very hot areas. So the winter areas are the winter times are more pleasant. So they want to have offsprings in the winter season. So you have these species from the cold climates that should give out their offsprings in the spring season, and these species that should have their offsprings in the winter season. Now what happened was that when we consider these crosses, so Capra ibex crossed with Hircus or with Nubiana, it rutted in autumn and it fawned in winter. So now remember, where were these species getting introduced and reintroduced? They were getting reintroduced in the high Tatra mountains, which are very cold areas. And when you had these individuals, Hircus and Nubiana, into that area, so the hybrids gave out their kids in those times where when this area was suffering from a very cold climate, because they were already because in their original climates they were having kids in the colder times. So basically what we are seeing here is that you have an ibex. Now ibex lives in cold areas, so it fawns in spring. Then you have Hircus and Nubiana. They live in warm areas, and so they fawn in winters. Now in the case of the hybrid, we had it living in cold areas and fawning in winters. So when the offsprings came out in winters and it was already a very cold area, so all the offsprings died out. So the offsprings perished from generation to generation in the severe winters, and ultimately we did not have a single individual left. So this is an example of outbreeding depression. The second example is that of roebuck. Now roebuck is also a deer like species, it is a herbivore species, very similar to our sambar deer. Now before First World War, the Siberian race of roebuck, Capreolus pyrargus, was introduced in Slovakia. Now Slovakia also had this roebuck population, but the roebucks of Slovakia were smaller in size, and the Siberian race is much larger than the Slovakian race. So when you had males of the Siberian race that mated with females of the Slovakian race, the fetus that was produced was so large that it could not come out of the orifice of the female, and so ultimately the fetus died within the body of the mother, and the mother also died subsequently. So no fawning was possible, and all the females died. So this is a case of physiological incompatibility between two of these races. So these are the examples of outbreeding depression, but now we will look at some more general terms. If you have decided that you want to translocate some individuals or a group of individuals from one area to another area, what are the planning requirements for this reintroduction? So what are the planning, preparation and release stage activities? So one is that you need to have all the approvals, so as we saw in our chapter on the legal aspects. Then you need to have a multidisciplinary team of experts, because this team of experts should know not only animal husbandry but also should consider the cases of transportation of these animals, the cases of immobilization of these animals and the feeding of these animals and so on. Then identification of success indicators, securing adequate funding, design of pre and post release monitoring programs. So when pre-release monitoring program would mean you need to monitor the animals before they are being released, and after they are released also you need to monitor them whether they are doing fine or not. Health and genetic screening of the release stock, vaccination of the release stock as required so that you do not bring in any diseases from one area to another area, quarantine arrangements as we saw in our lectures on disease monitoring, development of transport plans so that this transportation occurs in a very short time and without causing much trouble to the animals, determination of release strategy, whether you want to go for a hard release or a soft release. Now what is the difference? So you have a truck and you brought in animals into an area and you left them in the new forest. So this is a strategy that goes by the name of hard release. In the case of soft release, you have these animals, you bring them into an enclosure, you keep them in the enclosure for some time and then you release them into the forest. So this thing goes by the name of soft release. Now both have their own pros and cons. Now in the case of animals that have been chemically immobilized, we generally prefer to put them into an enclosure for some time so as to be able to monitor whether the impacts of the drugs have worn off. So essentially, if we had used say a narcotic into these animals, we need to be sure that these animals are not showing any of the impacts of narcotic drugs, and so we will keep them in an enclosure for some time, we will observe them for some time and then we will release them. Hard release on the other hand is used mostly in the case of animals that have been captured physically. So for example, in the case of animals that we captured using a BOMA technique. So BOMA technique was a mass capture technique that we saw in one of the previous lectures, and in that we create a large sized funnel into the forest areas using plastic sheets, and then we drive all the animals into this funnel. Once these animals are inside, we close these gates, then we drive them further. When they have all come into this area, we close these gates, and then there are some trucks waiting here, and all these animals move as a herd into this truck and then they are transferred to another place. So because these animals have been moved as a herd to reduce the amount of capture myopathy in these animals, we may go for a hard release. So in this case, we will directly bring this truck into the other forest and will release all these animals. But do you want to go for a hard release or do you want to go for a soft release is something that is very important during the planning, preparation and release stage. So you need to determine this release strategy because before you have begin your work. And then establishment of intervention policies. So intervention policies means that if you find anything that is going wrong, if you find any new diseases or these animals are not able to establish themselves, then what sorts of interventions are you going to put in? Are you going to say that will allow nature to as she wants, or you go with an intervention policy in which you say that we are going to micromanage everything? Then it also requires education, training, media coverage and involvement of the locals so that they are also attuned to our cause. And once you have released these animals, so once you have released these animals for introduction or reintroduction or supplementation, then we need to monitor all or a sample of individuals through tagging, telemetry, informants and so on. So for instance, when tigers were released in the Panna Tiger Reserve, then all of these tigers were fitted with radio collars. So in the case of those radio collars that are put in the neck region, so they continuously give out VHF signals, and then we had our departmental staff that were continuously monitoring these animals. So day in and day out they were seeing that these animals are surviving and that none of these animals have been poached, because of which our tiger population in Panna increased substantially. So we require to have a monitoring strategy of either a sample of individuals or of all the individuals, and ways include tagging, telemetry like radio telemetry and also informants. Then demographic, ecological and behavioral studies of the release stock, whether they are adjusting there or not. Then study of individual and population level adaptations, collection and investigation of mortalities. Now this is very important because in certain situations we have observed that you may have a very different species of plant that is growing up in the area where you are releasing the animals. Now this plant might have some toxins inside, and because your animals were never exposed to this plant, then it is possible that they would feed on this plant and then they would start dying off. So investigation of these mortalities becomes very important, and especially in the case of introductions of the species into an area where it was never found naturally. So this is also important. And then interventions when needed, including supplemental feeding and veterinary aid if something goes wrong, decisions to revise, reschedule or discontinue the program. So for instance, we observed this in the case of our Kanha Tiger Reserve. So we had a program for translocation of gores. So gores are Indian bisons, but then when they were being immobilized, we observed some amounts of fatality. So the animals died off. So in that case it was decided to stop the program then and there, so that we could look at it in greater detail, what were the problems and how we could improve our functioning. And once those were investigated, we had different plans, and then we were able to relocate the gores successfully. So you need to have these post release activities of revision, rescheduling or discontinuing of the program if you observe that these animals that were released, they are dying of capture myopathy, or they are dying because of the impacts of the drugs, or for any other reason. Then also habitat protection and restoration is important, because when you are putting in animals into a new area, then you need to ensure that there are no poachers nearby. Also public relations activities including education and mass media coverage, they are important. Evaluation of success and cost effectiveness of techniques used. Now this is extremely crucial, because any activity will not be a one off activity. If we have successfully translocated animals, then we might want to translocate more animals, and so after every translocation, after every release operation, it is necessary to evaluate the success and the cost effectiveness of techniques used, whether we can make any improvements for future. And also regular publications in scientific and popular literature, because after all we are doing this for conservation, we are doing this to increase our scientific knowledge, and so that other people are also able to use our technical expertise to translocate and conserve some other species that are found in their areas. So for that, scientific publications and also publications of popular interest articles becomes very important. So in this lecture we had a look at introductions, reintroductions, translocations and population supplementations. How do we differentiate between all of these? Why is it important? How do we reduce the amount of inbreeding through genetic rescue? How do we go for an outbreeding enhancement? And how in certain situations this, in place of getting an outbreeding enhancement, we could even get an outbreeding depression. So as in the case of the ibex or in the case of the roebuck. So those were two case studies that we saw in this class. And then we also had a look at what all things do we need to plan and prepare for whenever we are doing any reintroduction or any translocation. So that's all for today. Thank you for your attention. Jai Hind.